Welcome to the second lecture in the series Explainable Machine Learning, and today we will talk about how to look into a neural network. And after this lecture, you know how to visualize what was learned in the network and how you can, in detail, how you can visualize all these specific components in a neural network. But before we dive into today's lecture, let's have a look again at the general visualization of a neural network. So this is the, so this is the visualization I use for a neural network with these blocks, and this is also what is used throughout the literature most often when neural networks, especially convolutional neural networks, are visualized. So a neural network is nothing else than a chain of mathematical operations, and here denoted with F. Then you have the input image X, you have the parameters W, and you have an output Y. And so the neural network models the relationship between the input X and the output Y. The input is fed to the network with as input layer, which is in our case an image with RGB values. So one position in the input layer is an RGB pixel value. That means the dimension of the input layer is the height and the width of the image times 3 because we have RGB. The output is in our case an instance segmentation map, and an instance segmentation map is here you have at each pixel position a semantic class, but not only the semantic class, but also an index for the object in a specific class. So that's why for each object you have a different index. Okay, so the output in our case an instance segmentation, and so the dimension is the size of the image times 1, because you only need one channel to have the number of the instance in there. What is illustrated here in beige and in blue or turquoise, blue, green, this one are blocks. And these blocks, so one block illustrates the activation maps. That means the output after applying one or several of these mathematical operations. And these blocks are mostly just referred to as hidden layers, and these activation maps are a new representation of the data. And in the best case, this representation of the data is very suitable for this given task, more suitable than the input, which is in our case the RGB image. So you just try to learn a good new representation in a neural network. And if we illustrate such a cube in more detail, it would actually look like this here. So what you see is here are these small cells, and each cell in this cube is a single activation. And these two directions here are the position in the image, and this direction is the channel. For example, if you apply 4 one-dimensional convolutional filters to a one-dimensional image, so if you make a convolutional operation, you get 4 one-dimensional feature maps. So the channel number is 4. And you can also get other illustration. For example, here you can visualize a spatial activation. So these are all these are all the activation at one specific position, or you can visualize the channel activations. And channel activation are also called feature maps or activation maps. So throughout the lecture, I will sometimes use feature maps, sometimes I will use activation maps, but they are actually, the same. They are actually the same. So let's think about how we can look into a network. And within the last years, 2 big research directions has evolved to tackle this challenge. One is feature visualization, and the other one is feature attribution. And here they are listed with 2 examples. So for feature visualization, we focus on the question, what does a network look for? So here we select representative samples from the data set, or we generate samples, for example, or some examples which lead to high activations, or we visualize components in the network like the weights or the activation maps. And from this we get new insights, what was actually learned. The second one is feature attribution. Here we focus on the question which part of an example is responsible for the network behaving in a particular way. And here, for example, we highlight examples or parts of them. And here is a part highlighted, exactly this part of the fluke. And this I already talked about it in the last lecture about it. Today's lecture is about feature visualization, and I will also talk about these 3 points. And the next lecture focuses on feature attribution. So let's first have a look at how we can visualize model components. And here I want to focus on a use case, or actually a project we are concerned with in my team. And what we want to do is we want to discover wilderness characteristics. So on Earth, there are many well-defined land cover classes, but then there are land cover classes that are poorly defined, and one example is wilderness. So wilderness is nothing, so there are existing definitions, but they are not really technical, so it's hard to use them for machine learning. And what we want is we want to find out which appearances are typical for areas with no or little human influence. This is how what is one characteristics for wilderness. So wide areas have no or little human influence. And we want to find characteristics, patterns of wilderness in such areas, so that we can derive a definition that we can use for machine learning and mapping of wilderness. And for this, we use a specific data set. We defined the data set, or we collected data in the data set. And we used a study area, Fennoscandia. Fennoscandia is a huge area in Europe, and we used actually 2 classes. One class is the World Database on Protected Areas. So this is our, or short WDPA, and this is our assumption for now, or for this project, for this use case, that we assume that protected areas can be related to wilderness, because here we have nearly no human influence. And we have anthropogenic areas, and here we have, for example, artificial and agricultural surfaces. So all green areas in this image belong to the World Database on Protected Areas, and these purple areas are anthropogenic areas. Okay, so what we used are Sentinel-2 images, that means satellite images. And here you can see some examples of such satellite images. And these are a few examples from anthropogenic areas and protected areas. What you can already see is that, trees and green is not only typical for protected areas. So also the anthropogenic areas are quite green. They have trees in there. So it's not the only appearance for it was our first thought, and our first thought was just not correct. And we used image patches from these larger satellite images for analysis. And to distinguish between these 2 classes, protected area and anthropogenic areas, we use a network called so-called jungle net. And this network is a combination of a UNet and a convolutional neural network and a fully connected network. So UNet is illustrated here in this blue-green, and in beige is the convolutional neural network and the fully connected network. And we use this network for scene classification or image categorization. That means you have given a scene and the output is the score that the scene belongs to a specific class. In case we have only two classes, this is the case for us, or in general, if you have only two classes, we can assume that both classes score to 1, or the scores of these two classes sum to 1, and then we only need a single output score, because otherwise it would be redundant. And this is also how we model this. The good thing about this network is that we can have a look at the activation maps in different locations in the network, but also after the unit. That means here in the last unit layer. And of course we designed the network to that it does it like this. And so here the activation maps have the same height and the width as the input image, which makes it especially interesting because it contains a lot of information. So we can easily analyze it, but this layer is also used for decision making. So the actual more decision making part is done here. And the unit is the main task of the unit is not only the decision making, but learning a good representation of the data, which tells us something, gives us new insights. And in order to make an actual decision, so there we use this CNN here to have a classifier. And the whole network is optimized so that we can make a good decision. And during the process, the activation maps are determined. And now a good question is, what can we actually visualize? So we can visualize different components in the network. And so we have different possibilities. One are the activations and the other one are the weights and the filters. So when we visualize intermediate activations, we focus on the question what the network saw. That means what was extracted from the input. To repeat or to say it again, so the activations are the output after a specific operation. So we simply plot what the operation has produced. And then we can visualize weights and filters. And here we focus on the outputs are patterns each layer has learned to extract from the input. It's a little bit different. So the weights and the activations. So the weights are actually what is used to compute an output. So what is used to compute the activations. And so this visualization of weights and filters is generally used for convolutions. Because here the weights, the filters are learned. So these filters are weights and they are spatially arranged. So that's nice for visualization. And what I can already tell is that activations are much easier to study than weights. So this is actually my impression. So maybe you have a different impression after I show you some images, but this is actually what I think. Okay, so let's have a look. No, first let's have a quick reminder what actually is happening and what causes high activations. So what you can see here, so this is a satellite image from the desert in Saudi Arabia, and all these circles are agriculture fields. So this is because of a specific irrigation system called pivot irrigation, and that's why they have such round agriculture fields. And we only use for simplicity now one channel. And here you have a filter, and you can already see the filter has a similar pattern as you can find in the image. So when we apply learned filters in a neural network, we get high values. Exactly here, high values are bright, not bright colors, but white means high values and black means low values. And we get high values when the filter shows similar pattern to the image, and if the filter has also high value. Because normally in a neural network, you just multiply the filter with the input and with an optional normalization. But this is actually what is happening here. So the bright colors you see is actually exactly the position where the filter has a similar pattern as the input image. So let's have a look at the weights. And so here visualized are several weights. All the weights, as is typically done in a neural network, are small. So they have a size of 3 × 3. I will tell later also a reason why we normally only use very small filters in neural networks. And what you can see is that you might see that you have basic features. Sometimes you have more homogeneous regions. Sometimes you have more horizontal structures or diagonal structures. Yeah. Then if we go deeper in the network, so this is the 4th layer, what you can see is quite similar. So you have some weights, again 3 × 3, and you can see sometimes more homogeneous features, or you can see some here, for example, maybe a bit of a checkerboard or a diagonal structure, and yeah, and so on, similar to the 1st layer. If we go deeper, and in this case we made the filters a little bit larger, in this case 5 × 5, what you can see is it's more complex. So it's not these basic structures anymore, but what you already realize is that it's not immediately clear how to get insights from the weight. Actually, I don't, or for me it's difficult to see. Yeah. And now you might ask, so why is it so difficult to get something out of it? So first of all, there is a lack of contextualization. That means the visualization is meaningful, or only meaningful, when linked to an input, such as RGB values. So actually that means the weights are only really meaningful when you're on the 1st layer. Because in the 1st layer, when we visualize the weights, they are linked to the RGB values, and this is something we understand. So if we learn diagonal structure, or the network learns that there are diagonal structures, we can assume that in the input image there are diagonal structures. And due to this connection, these weights in the 1st layer are meaningful. But weights between hidden layers are less meaningful because we don't know anything about the source. And even if we see the source, that means the activation in the previous layer, this is nothing we can make sense of, because the activations are complicated, plus weights are hard to see. This is very, very abstract, so it's hard to make sense out of it. Then there's the problem of indirect interactions. So sometimes there are meaningful connections and important weight connections between the neurons which aren't directly connected in a neural network. So for example, in a residual network, some neurons are copied and interact with deeper layers, or layers in later networks, with neurons in later layers. With all these skip connection, or every time when there are some interactions in the network where neurons are not immediately connected, it's very hard to make sense out of it and to grasp what's actually going on. And then there's the dimensionality and scale problem. So generally the number of weights is very high, so therefore the visualization is super overwhelming. Like for example here and here, this is only a part of all the filters which were actually learned. So yeah, So if we plot all of them, it gets even more overwhelming. So let's go away from the weights and let's have a look at the intermediate activation. Let's come back again to our projected versus anthropogenic data set. And so here are two two images in the first column from our data set. And what you can see here are the activation maps in a shallow layer, in the bottleneck layer, and in the deep layer here. So what you can already see is that in the shallow layer, you have more fine-grained structures, and in the deeper layer, it seems nearly that the activation's a little bit connected to semantic concepts like a lake, or it's actually they focus on bigger structures. And what you can also see is in the bottleneck layer is very coarse, because of course in the bottleneck layer you reduce the dimension of the activation maps, and in order to relate it to the original image, you need to upscale it again. Yeah, so this is also summarized here. So what I already said, that earlier layers focus on general features like edges, and deeper layers focus on more complex things, but only looking at the activation maps is here it gets difficult to interpret visually. The reason is that the network takes the visual information of the input image and tries to convert it to the output classification domain. That means it gets very abstract in the intermediate layers, which patterns are actually important. And there's also one thing, only important information is retained, and this is so-called information distillation. So yeah, the network compresses a lot. It gets compressed and abstract. And then you can, we have something like blank activation. That means activation maps with a lot of zeros, like for example this one here. And this indicates that these features could not be found in the input. Okay, the reason why we have these finer grained structures in the first layer or in early layers, and in deeper layers we have more complex structures, and also the reason why we normally only use small filters is the receptive field. So most of the network has this typical encoder-decoder structure. That means you downsample, and if the filter size stays the same, which is illustrated here and which is normally the case, but the images and the activation maps are actually downscaled, the receptive field, that means the area from the image what the filter sees, gets larger. So the receptive field size increases, and this can be seen here. So even if the filter is very small, if it's downscaled, it sees a lot of the image, and or not immediately from the image, but from the activation maps which were extracted from the image. And so the activation in deeper layers capture more spatial information. And so let's come back to our JungleNet. I mentioned already that the last layer in the unit is particularly interesting. So why is it so? First of all, it has the same size as the input image. So we don't need to upscale, and the information content is quite high. And furthermore, it's a deep layer. So this is already a layer where the representation is quite complex, and it should help a lot for decision making. So it contains a lot of information, and it has the same size. So it can be assumed that it's very informative for us, and it contains a lot of informative features. So let's have a look what these maps can tell us. So what you see here, so first of all, we use our trained network and derive 3 activation maps. This is an important information. Since we have 3 activation maps, we can visualize it as RGB image. So of course you can use less activation maps and more activation maps, and then you can just visualize them as single channels. And here you can see a few of these training samples from our data sets, and for all of these training samples we compute the activation maps. And here they're illustrated as RGB in RGB. We can already see that water produces pink colors, and if we think about RGB channels, that means it has high values in the first and the third activation map, but low values in the second activation map. We see that where land has more yellow colors, that means it has high activations in the first and the second activation maps, and we can see here that anthropogenic areas are more blue. And when we now put every pixel in these activation maps into a 3-dimensional space, we get a visualization of the activation space, which is a really nice, I think really nice visualization, because this tells us already how variable the activations are, and or if we have, for example, specific clusters, and this might be good for classification. And in the end, it gives us already an impression on how distinctive the patterns of the objects and the areas in the image are. So you can see it doesn't look like an exploded pillow. You see a lot of structure in there. And there's another interesting thing we can do. What we can do is we can connect the activation space to other data, for example, label information. And so here, what you can see here is a patch from the so-called Korean dataset together with the satellite image and the activation map. So Korean is a land cover dataset for whole Europe. With a ground resolution of 100 meter and 44 different land cover classes, and it's updated every 6 years. So it has some interesting information for us. And by connecting the activations with the land cover classes, we can derive an enhanced activation space with land cover information. And this can be derived, for example, by assigning each activation in the activation space to the land cover, which is most often assigned to exactly this activation. And I want to mention again that this information can be derived even if the network was not trained for it. So we trained it for scene classification, but such information can be derived if you afterwards combine it with some auxiliary data or some other kind of information you have. And so it shows that the activations carry useful semantic information. So it was not trained for it, but it shows a clear connection to these labels. So it carries useful semantic information. So let's come to the next part of the lecture, which is feature visualization by selecting dataset samples and generating examples. So first of all, let's dive a bit deeper into which patterns can actually be used for visualization. So the smallest unit is a neuron, and so the neuron can be seen here. This is one activation. So with neuron, I mean one position in an activation map. In a convolutional neural network, this would be the output of a convolution of an image, patch, and a filter. Both of the same size. And what else? We can look at the channels, and that means we can have a look at the activation maps. So something in a similar way we did before. And in a convolutional neural network, this is the output of one convolution in an activation map in a previous layer and one filter. And then we can have a look at whole layers here, which is nothing else than looking at all activation maps together. And this is what is used in Google's Deep Dream, which I explain later how this works. So, but let's first have a look at feature visualization by selecting dataset samples. So when we want to learn which patterns were learned in the network, we can utilize a dataset, either the training samples or some other kind of data, but with the fixed network. So what we can do is we can select samples from the dataset that maximize a specific activation or a set of activation. So let's have a look at this equation, because this is exactly what is happening with this simple equation. Imagine you're at a specific position in the network. That means at a specific neuron. Then A, this here, A is the function that will deliver the output after or at this specific position. So afterwards you get one activation, and this is what is interesting for us. So what we are searching for is a data sample from the dataset. So X should belong to the dataset, and so we are searching for a data sample from the dataset which results in a high activation. And so this is actually what is meant here. So we use the argument, the argmax function, and we search for a data sample which actually maximizes this here. This can also be extended to maps and layers. We can do this by summing over the activations, for example, in a specific in the specific layer, so over the whole layer, or in a specific channel. Or you can even use it for groups of neurons. It's very easy, you just take the sum. And so when we use our jungle network, we could also look at specific positions here, and we actually plotted here 3 images from one specific activation. So these 3 images cause a high activation in a specific specific position in the network. In our case, this is in the second part, in the classifier block, the CNN, the 11th layer, and the neuron 30. And this specific neuron is, it seems so, firing when he sees water bodies. So the activation is maximized when there's a big water body in the image. And this, of course, you can do with a lot of different positions in the network. I will later mention it again, but with another data set. The advantages are clear. It's super easy. You just search for the images which cause a specific activation to be at the maximum, and the samples are realistic because they're actual data samples. The disadvantage is it's unclear which elements of the sample cause the high activation. So for us here, it was quite clear, but imagine you look at activation, for example, here on in previous layers. So it's not really clear. Or sometimes the images can still look quite different, but they all cause the same activation to be high. It's especially the problem in early layers when the filters and the activations are fine-grained, and the filters only look at the basic structures, or they resemble basic structures. So in the next part of the lecture, I want to talk about Deconvnets. Deconvnets are a simple visualization technique to visualize activation maps. And so deconvolutional neural networks, or short Deconvnets, were proposed by Seiler et al. Already a few years ago. And the Deconvnet can be thought as as a convolutional neural network that uses the same components like filtering or pooling, but in reverse. So instead of mapping pixels to features, Deconvnets project the feature activations, that means the convolutional outputs, back to the input pixel space so they can be visualized there. So we backpropagate the activations instead of the gradients, which is normally used as a step to, or the procedure to train the network, to optimize the network. But here we backpropagate the activations. So what is actually visualized then are the input patterns that cause specific activations. And if you think this together on the context of convolutional layers, then single convolutional filters are analyzed by utilizing what they have produced in their associated activation map. But let's have a look at it. So to understand Deconvnets, let's have a look at a simple convolutional neural network. A simple convolutional neural network uses convolutions, ReLU, and max pooling as a standard block. So here, this whole thing is a standard block with these three operations in a convolutional neural network, and we denote in the following one layer as a set of exactly these operations. So here's a simplified illustration with only one channel. That means a green block. And so what we do first is we have a convolution. Then we apply ReLU. That means nothing else than just cutting off all the negative values. Then we have a max pooling, which reduces the size by compressing the information. And so this can be seen as one layer in the convnet. What a Deconvnet now does is that it designs a block which performs the reverse operations. So namely, we have a max unpooling, we have a ReLU, it's named the same because it's exactly the same. And then we have a deconvolution. And so these are all the reverse operations. And as you can see here, so from this block, if you perform the reverse operation, you can actually go back to the input in the network. So let's have a look at the procedure. First of all, what you do is you forward propagate an image. And then you choose a filter of interest. So something, let's say you want to learn something of a specific filter. Then you set all activation maps, that means all channels, to 0 except the activation map which belongs exactly to this filter of interest. And then you attach a Deconvnet to each layer and follow the path back to the input space. And then you visualize the input to show the pattern the activation of interest is sensitive to. So let's dive a little bit more into this reverse operations. So let's first have a look at unpooling. So here, let's first have a look at pooling operations. So first of all, when you have an image like this here, and you perform a pooling, in this case a max pooling, what you do is you have so you have a specific window size, in this case of 2 × 2, and from the first few values in the upper left corner, you take the maximum value in this 2 × 2 window, and this is here the one. Then you have a stride of 2, and the maximum value in the next part is 3, and the maximum value in this upper in the lower left corner, the maximum value is 2, and if you shift it again by stride 2, the maximum value is 4 here. So this is the max pooling. In this case, we have a window size of 2 times 2 and a stride of 2. Of course, there are other parameters, and of course, I refer you to, if you want to know more about this pooling and how to actually perform it and all the different pooling. There's a lot of literature out there. But now the interesting thing is the max unpooling. So for the max unpooling, what you do is, first of all, you need to store the max values and the positions in order to go back, to reverse this. So it's not only that you have this information, but you also remember where it was. And all the other positions are just filled with zeros. So here, before, you see there were a lot more, but this is the information which is kept, and also the position, and this is what you can restore. So this is the reverse operation. And then there's before there's a ReLU operation. I already said that ReLU is exactly the same. So the reverse operation of a ReLU is again ReLU. So if you would have here negative values, what you do is you apply ReLU and all negative values are set to 0. So deconvolution is a bit more complicated, and we need to be aware that there's not such an operation like a reversed or inverted convolution that calculates the former input to the convolution. It's the same with the unpooling. It's a reverse operation, but it will not really restore what was there before. For deconvolution, you use the learned filters from the operation you want to reverse. And then you horizontally and vertically flip the filter. Why this is actually done, I will show you in a few seconds. So let's have a look at a specific example. So what you have here is you have an activation map X. For example, this is the one after the unpooling, and W is the learned filter. But before going through the steps, an important point is, very important in this context, is that neural networks normally implement cross-correlation, not convolution. Cross-correlation and convolution are both operations which are applied to images, but cross-correlation, so cross-correlation means sliding a filter over the image and then do something like multiplying and summing or do something. And convolution means sliding a flipped kernel across an image. So most convolutional neural networks and machine learning libraries are actually implemented using cross-correlation. And in the end, it doesn't matter because it will not change the result. In practice, if we would use convolution instead of cross-correlation, it would mean the same weight values would be learned, but just in a flipped orientation. But let's have a look what would happen if we would not flip the filter. Again, what you see here is a feature map X, and this is the filter, not flipped. First, we extend our activation map so that we do not have some border effects, and so that we get exactly the same size afterwards. So let's perform now the same operation as it's usually done in a neural network. So we take you have the filter, and you have the feature map, and you put over it, you multiply it, and this is the outcome. So it's all 0, so the result is also 0. Again, if you shift it by 1, it's 0. So not so. If you perform it in this way, not so super interesting. But now it gets interesting at this position. So here you have a 1 at exactly where the I is. So in the end you have an, when you apply it, you have an I. And if you shift it more, you have an H at the position 1 and so on. And if you perform it in the sliding window approach, and you do, this is actually the result. And what you see is that the result after applying the filter to this activation map is a result with the pattern of a flipped kernel. And this is not what you want. What you want is, you want, if you have one single activation, one in the image, in the activation map, and you apply a filter, you want exactly the same pattern afterwards. So in this case, after the deconvolution. That's why you vertically, horizontally and vertically flip. That means you rotate the filter by 100 degrees. And if you do this, this is actually the result. And this is, so this is what you want. You have the original filter, this one, rotated, and this is the result. So you will exactly get the pattern of the filter. Okay, so let's have a look at some visualizations, and here I will take the examples from the paper from Seiler and Fergus. And so let's have a look at a few example results. Let's say you trained a convolutional neural network, a simple one with some blocks as the one I showed you before, with the cont, the ReLU, and the max pooling, and you trained it on ImageNet data. And so again, or I want to repeat it, what we already did, because this is also useful as a visualization and for comparison in this context. You can scan through your data set to find image patches that maximize a single activation in the first layer. So let's focus for now on the first layer. And here, since the first layer anyway only see a small patch of an image with the size of the receptive field, that means in this case the filter size in the first layer. Here we plot only the part of the images of the image which influences the actual activation. So here, each of these, you have these 3x3 blocks. Each of the 3x3 blocks are image patches which cause one neuron in the first layer to be highly activated. For example, you have what you can see are diagonal patterns, for example in the upper left corner, or you have green homogeneous areas here in the middle, on the bottom, or you have orange patterns and so on. So the units in the layer 1 look at basic patterns. Again, we can confirm this. So, but what is actually interesting in the context of Deconvnet, because this is something we already know from, or I already told you about it, is this here. So these are, in the bottom you see the Deconvnet reconstructions. So for example, you choose an image patch from the top left group of the image patches where the arrow there points at. And then you proceed with the Deconvnet reconstruction, so reverse operations, and then you obtain such an image patch where the lower arrow points at. So this is here in this illustration, this is actually this one here, the upper left, the one reconstructed image patch in the upper left corner. And of course you can do this also to the others. For example, here for the green image patches which cause the same neuron to highly activate, this is the reconstruct. If you choose one of it, this is the reconstructed image patch with all this Deconvnet. Okay, this can be done for other layers as well. Here again you see on the right, so here you see groups of 9 image patches, here for example, and here. So you see 9 image patches which cause a maximum activation in the second layer. So now we look at the second layer, and on the left you see the images which are produced by the Deconvnet. So remember, you can use one image, you pass it through the network, and then you can make all reverse. And these are image patches which cause high activation, and this is when you use them for actually reconstructing and to visualize what the network has learned at specific points in the network, specific positions in the network. In the middle, for example, you can see on the middle of all these visualizations, you see circular structures, or you can see horizontal line patterns on the bottom, on the top left, or some stripe patterns and so on. I think it's really, this is really nice to see what's actually learned. Okay. And you can do it also for more layers. And here you can see, for example, some more complex structures such as honeycomb structures on the very left. Or here you can, it gets quite colorful. You can see the back of specific objects and so on. And again, because the receptive field gets bigger, you get more complex structures in deeper layers. Yeah, and if you go to really deep layers, you can nearly get objects. You can nearly, for example, see here. This is nearly This is nearly a dog detector. So because it all dog pictures of the same kind cause one activation to highly activate, and if you take this or if you use the Deconvnet approach, you can see these dog features, dog patterns which were reconstructed. Yeah, so that was Deconvnet. But now let's, so far we only used quite simple method how you can visualize single components or Deconvnet. But there's another powerful method, which is feature visualization by optimization. So neural networks are differentiable with respect to their inputs. So if we want to find out what kind of input would cause a certain behavior, like a perfect classification score, or that a specific neuron should be activated as high as possible, we can use derivatives to iteratively manipulate our input to the goal. And this is called feature visualization by optimization. And in detail, we formulate the following optimization procedure. So what we want to find here is an image X that maximizes the activation A here of a neuron with the index I and in a given layer L. So let's assume we have indexed all the activation into a specific layer. And this can be, for example, a classification network. And instead of minimizing the loss function now, we maximize the activations. And this is so-called activation maximization, as the name says. And yeah. And this can be extended to groups of neurons such as channels and layers. So I mentioned it already a little bit. And here we make it a little bit more explicit. So here you can also see the indices R, C, and B. So R and C are the row and the column, and B is the channel. And for example, you can perform activation maximization over the whole channel. So you need to sum over a row and a column in a specific channel. or you need to sum over the row and column and the channel, and then you have here a specific you perform activation maximization in a layer, and this is exactly what Deep Dream does. So what question can we now answer with activation maximization? So first of all, the analyzed neurons can be in each position of the network. And depending on where they are, it will answer different questions. So we can, for example, ask what are typical low-level or intermediate patterns the network has learned. Or we can, look immediately before the decision layer, and then it will answer what is a typical example for a specific class, a so-called prototype. So it will answer the question, what does the network think is a specific class? So let's have a little bit more, let's dive a little bit more in the optimization process. So it's a non-convex optimization problem. And so first of I need to mention for the following, we omit the indices. So imagine A is a selected activation you want to analyze somewhere in the network. And then we perform gradient ascent, which is similar to gradient descent process that is used to train neural networks during this backpropagation process. The difference is that here we are optimizing the network input instead of the network parameters W. But here, so we are not optimizing the parameters, the whole network is frozen. So, and what is actually, what you can see here, so this is the gradient ascent step with the step size, and T is the iteration. And we stop as soon as the neural activation has reached a desired threshold, because the activation increase when the image change, and so you can just stop after a while, or a certain number of steps has passed. So again, so this is the equation for the update step, and first of all, what you do is you select a neuron, so specific activation of interest, or of course you can extend it to channel and layers, but for now let's focus on a single activation. So you select one, then you perform a random initialization, which is XY. That means, for example, just a noise input, a noise image, a random image. Then you compute the gradients while the network is fixed. You perform a gradient ascent update step in small steps, and then you iterate the steps 3 and 4 until the gradient reaches a threshold or fixed number of iterations. And so you will reach a specific point at end result. And you can increase the diversity so that you do not have one output. You can increase the diversity between these generated examples, these activation maximization samples, by using different seed points for this random initialization here. But there's one challenge now. It sounds really easy, and what I can already tell, this is a basic concept. But the basic concept is not really well working in practice. This is because it very often leads to unrealistic, that means uninterpretable results. So without conditions, so some boundary conditions, how this image is actually generated, there are too many solutions. And there are so many solutions which cause high activation, especially in a high dimensional space, and most of these solutions make no sense. So there are so-called fooling examples, and this is when the pattern has a high frequency noise. So high frequency noise is when it's not when the image is blurry, it's more like it has a fine texture, so fine noise everywhere. Salt and pepper, very fine, but nothing what you want. But it very often causes this high frequency noise. And activation maximization initialized with a real image can turn into a so-called adversarial examples. And this is an adversarial example, is a realistic example which causes an unexpected and mostly undesirable behavior of a neural network. And I will show you one example of such an adversarial neural network, such an adversarial example. Again, we used our jungle network and to compute an activation map. So what you can see here, you have an image, and this is the activation map in the RGB, or illustrated as RGB, these 3 activation maps in the last layer of the UNet. And we identified that everything what is illustrated in yellow can be related to patterns which occur in areas with less human influence. So what we did is we want to change and influence the image. We want to make it, we want to turn it into more protected, more wide patterns. So what we did is we maximized the activation assigns here to one part in the image. So we said, okay, this corner should be more wired. But what happened is after activation maximization, nearly nothing. So if you look very closely, you can see this high frequency noise, but still you can still see this urban area. And this is a so-called adversarial example because it will cause a much more higher score overall, also for the classification for protected area, and it will show wide characteristics. But actually it's, for us it looks like urban, it's still urban. And in order to overcome this challenge and to tackle this, these adversarial examples, we can guide the optimization more towards natural images. And that means we incorporate natural image priors into the objective function, into the optimization process, which was found to be substantially improve the recognizability of these images. So to give you an example, so here again, these the example of this agriculture field in Saudi Arabia, and natural images are, for example, smooth and they barely have extreme intensities. And this can be seen, for example, when all these RGB values are illustrated in an RGB feature space. It's not that the whole feature space is filled with, it's mostly along the diagonal, and this is something what natural images, they are mostly like this. So very smooth, no abrupt changes in images, and barely extreme intensities. And this knowledge, this prior knowledge, can be used to guide the optimization process. So for example, an image prior may encourage smoothness or penalize pixels of extreme intensities. And such constraints are often incorporated into this activation maximization formulation as a regularization term. And this regularization term is R. And so the update rule then is, this update rule here, it's extended by the regularization term. So what we have is we have X, which is the current input image. Then we have a term that causes the activation to be maximized, and then we have a term that causes the regularization loss to be minimized. And so this is what we've done before, and this is a better guidance. For example, to encourage the smoothness in an activation maximization image, we can use the total variation. Total variation means we or that means we introduce a total variation loss, and total variation is the sum of the absolute differences of neighboring pixel values. And of course this should be low, and then you have a smooth image. This is something we want. However, so in practice we do not always compute actually this term here. So there's a trick, something very simple we can do. So we do not compute the analytical gradient, but instead we utilize a Gaussian blur kernel. So Gaussian blur kernel is also something which causes smoothness. So in every step, in every iteration step here, we blur the current image, we blur the current image a bit and continue with the algorithm, which is a very fast implementation of such a regularization, such an image prior. And there are a lot of such regularization terms, and nowadays activation maximization is actually not used anymore without them, because it would just not, in its basic form, it would just not produce good results. So let's come to a very famous procedure called deep dream. So deep dream, here all activation in an entire layer. So here we look at an entire layer, are maximized instead of a single activation. So we do not look at a single filter, a single activation. We look at a whole layer in the middle of the network, or in a more complex layer, in a deeper layer. And the initialization is done here with an image instead of a random noise. This is one crucial thing, one essential thing for this deep dream algorithm. So when we go through this deep dream algorithm, another huge difference to the methods I showed before is that deep dream uses so-called octaves. And that means we apply the gradient ascent update rule in this computation on different scales. That means we do not use only the original image, but also smaller versions of the image. And this helps to reduce the noise, but also it helps to increase the granularity of the patterns. So when you have a small image and you apply this procedure, you get bigger bigger differences, and if you have a huge image, it will cause that you have smaller patterns, which are caused by the activation maximization procedure, and so on. So you have on all levels, on all scales, you work with it. So let's go through this procedure. So first of all, you choose an image and you define the number of scales what you want to have. So the number of these processing scales. Here I illustrated 3 of them, and then you resize the image to the smallest scale. So actually you start working with this image here. Okay, so in the next step you perform then gradient ascent and you upscale the image to the next scale. So here the whole algorithm is illustrated. So what you do first is you downscale, that's what I showed before, and you start with this image and you apply deep dream algorithm. That means you perform activation maximization for the whole layer. And then there's another step which is crucial here. You re-inject the details which are lost during the upscaling. So this is here, your upscale, so you go to the next scale. The thing is, if you downscale and then you upscale again, there are details lost, and this is actually the point where the information of the from the multiple scales are combined, and it should not yeah, it should not suffer from lost information due to the downscaling. And to obtain the detail, to re-inject the lost details during the upscaling, what you do is you simply take the original image, shrink it down, and then you upscale it and then compare the result to the image before the downscaling. So you actually, when you downscale and then you upscale again and you compare it, you actually see what was lost. And this lost information is re-injected here again. So you can work on all scales, but yeah, with limited information loss. And repeat step 3 and 4 and stop after the larger scale, so the original size is reached. So to summarize this, so on all scales you perform deep dream, you start on the smallest scale, and you re-inject the lost information due to the downscaling and upscaling procedure here and here again. Okay, and then all the due to this the information due to the activation maximization is transported from the smaller scale to the larger scale and combined. And when you think about it, it's all nice, but the difference for deep dream is also that you start with not with a random image, but with a given image. And what is actually happening when you do it is it results. So the features in the input are amplified with what was learned in the network. So therefore, the result depends on the input and what the network has learned. So it means the output depends on the features in the training data. And here you can see one example. So this is one satellite image from an area in Switzerland, and this is when you perform deep dream with a pre-trained network on ImageNet. And remember, ImageNet contains all these huge amount of classes of natural objects, animals, and so on. And this is when you use a very early layer. And what we learned today was already that in early layers, basic structures are learned. And what is happening here is the deep dream algorithm amplifies all the features which were learned in the network. That means these edge-like structures, and this is all amplified. And you can also see that the basic structure, the basic image is still there. It's just yeah, very psychedelic. And if you perform this algorithm on later layers, it gets a little bit creepy, I would say. So this is from an intermediate layer, and this is from an even later layer. And what you can see here, this is happening very often, also with other images, which you can see in the literature, that eyes appear. So if you have maybe somehow some circular structures in the input image, this is amplified, and it turns into eyes. Or like this lake is somehow a little bit like, I don't know, legs or something, because the data set is more about animals, humans, and objects that surround us. Maybe something like this. Maybe you have even better ideas what can be seen there. Yeah, so very interesting. The thing is, if we would use another trained network, this would result in different images, because then different kind of patterns would be amplified. But what does it tell us? This image now can be, first of all, we can see what was in the image, which patterns were in the image, but also what was learned by the network. For example, these round structures and yeah, these legs or whatever. And also interesting thing is here, so this is, you clearly see basic structures, and here you see more complex structures. To sum up, so different components in a neural network can be visualized where activations are more intuitive than ways. This is what I showed you in the first part of the lecture. Then we talked about activation maximization, and activation maximization, so the basic principle behind it is actually the basic for many visualization approaches to understand which patterns a network has learned. But a pre-trained network is necessary. So all these techniques I showed you today, except the Deconvnet and what I showed you in the beginning, but these activation maximization approaches, they rely on the pre-trained network, and it's really hard to train a network on scientific data. I already mentioned it in the last lecture, because we do not have so many data that we can train a large network. Yeah, and also, I mean, these here for ImageNet, it has very clear structures, but for example, for the wilderness task or the wilderness project, it turns out to be really complicated to produce such results. Okay, but that means a lot of open research questions, a lot of interesting things we still can have a look at. And I want to mention also further literature. So as always, you see references in the bottom of the slide. So every time I present to you an algorithm, the reference on the bottom. And I can highly recommend Distill, which is an online journal, and here a lot of really nice papers and source code related to feature visualization is presented, so I can highly recommend it. I used it also for the preparation of this lecture. And with this, I thank you very much, and I hope we see each other in the next lecture, which is about all these saliency maps and heat mapping, and which will gives you the other research direction besides the feature visualization. Thanks a lot, and see you soon.